Today we'll look at the structure of FIBSC syllabus. As you all know that this is the autonomous, we have obtained an autonomous status. So this is the structure, core structure for FIBSC microbiology autonomous, which is a choice based credit system. So we need to understand, you need to understand what is this credit system. You see that this is the credit system which is designed for FIBSC. We can see the second column, that is class FIBSC. So this is all structure for FIBSC. In third column it is mentioned that semester number. So you see that there are two semesters, first semester and second semester. This means that this will be a semester pattern. That means at the end of each semester you will have to give examination. Okay, so the assessment will be done at the end of each semester. Assessment will be of two types: internal assessment and external assessment. So you can see the last column, that is 50 plus 50. That means 50 marks will be there for internal assessment, and internal assessment will be based on your different tutorials, your attendance, your behavior, etc, etc. And there will be 50 marks for external examination. So that will be conducted at the end of each semester. Now you see in this table column number 4, that is code, that is a subject code: micro 1101, micro 1102. So micro 1101 and 1102 will be the two theory papers for first semester. Okay, so likewise in first semester you will have to go for performance of one practical, that is micro 1103. So in all there will be three papers, two will be theory papers, one will be practical paper in semester 1, and likewise in semester 2 similar pattern will be there, two theory papers and one practical paper. So the title of paper is mentioned in sixth column. You can see that. Now this is a credit system. You see the last but one first column, that is seventh column, there is a column of credit. So for each paper there will be 2 credits. So you see, for theory paper 1 in first semester there will be 2 credits, second paper 2 credits, and practical paper 2 credits. So in all how many credits are there? 12 credits are there. So it means that you need to complete 12 credits in order to get through FIBSC. Okay, so this is in brief the credit structure. Each credit will be consisted of 18 lectures, 1/8. Each credit will consist of 18 lectures. So that means for semester in semester number 1, for paper 1, that is micro 1101, there are 2 credits. That means 18 into 2. So in all 36 lectures will be there for paper 1, and likewise there will be lectures for paper 2 and practical examination, practical course also. This is the syllabus for semester 1, which for paper 1, that is micro 1101, semester 1. As I said earlier, that it consists of 2 credits. So in all there will be 36 lectures. Okay, so you see this table, the last column, it mentions about the number of lectures. Why number of lectures are important? Because this will tell you the weightage of that particular topic. So if the number of lectures are more, then there will be a more weightage. This means that there will be more number of questions asked on that particular topic. So you see the first column, credit 1, then second column is of topics, third column is of lectures. So you see credit 1, there are 3 units: applications of microbiology. So applications of scope and applications of microbiology, this topic will going to explain you regarding this, regarding the importance and in how many different types of fields are there in microbiology. So scope of microbiology is going to be one of the very important topics in order to understand the importance of this subject. Unit 2 will be focused on the applications of microorganisms, particularly in relation to agricultural microbiology and medical microbiology. So these two will be just for the sake of examples. Unit 3 is focused on morphological and differentiating characteristics of microorganisms. So we'll see the different classification systems of microorganisms, different examples, different categories, and general characteristics of these microorganisms along with their examples. The credit number 2 consists of Unit 1, where we'll try to understand the different historical developments in the field of microbiology, different contributors, significant contributors, experiments performed by different scientists. Unit 2 in second credit, it will be development of microbiology in 19th century. This is in continuation with the history of microbiology part. So advanced developments will study under this unit 2 in relation to for development of fermentation, surgical antisepsis, germ theory of disease, etc. And unit 3 will consist of, in continuation to the history part, the latest development, that is in 20th and 21st century, what important developments had taken place in microbiology that will understand. In semester 2, that is micro 1201, this is theory paper. Paper 1, again there will be 2 credits. The title is same, Introduction to Microbiology. There will be again 2 credits, credit 3 and credit 4. So in credit 3, there are units. Unit number 1, basic biochemistry for microbiology. So understanding chemistry is very important in microbiology because there is a separate branch in microbiology. We call it as microbial biochemistry. So in order to understand biological chemistry, we need to first understand the basic concepts in chemistry. So the basic concepts, you might have learned them earlier, like definitions of atoms, elements, ions, and molecules, regarding molarity, normality, isotopes, Avogadro's number. I hope you must have learned this in your school days or in 11th and 12th. So this will be just a revision for that. Different types of bonds, like covalent bond formation, then covalent bond formation, then ionic bond formation, and hydrogen bond formation. Then biochemical reactions, the concepts of metabolism, catabolism, anabolism, the role of enzymes, concepts of pH, acid, base, strong acid, weak acid, base, buffers, etc., etc. So these basic concepts we are going to learn in this particular chapter unit. Second unit will be based on, which is in continuation with biochemistry, is the structure and functions of biomolecules. So basically there are 4 types of biomolecules, which what we understand are carbohydrates, lipids, proteins, and nucleic acids. So in general, we'll be looking at the structure and functions of these 4 different types of biomolecules in the cell. And the fourth credit will be consisted of one unit that will be mainly focused on bacterial cytology. So you see that microbiology, study of microbiology is dominated by bacterial metabolism, bacterial genetics, so most of the studies are focused on bacteria as a model for the study of microbiology. So it is essential to understand bacterial cytology. Cytology means the study of structural components of a bacterial cell. There are of course different types of bacteria having different shapes, different morphologies, different characteristics. We'll study in general a typical structure of a typical bacterial cell and its components. So these components are listed here. These are structure and functions of flagella, structure and functions of fimbriae, pili, capsule, cell wall, cell membrane, endospores, ribosomes, chromosomes, extra chromosomal DNA, cell inclusions, etc. So we'll be studying the structure and function of these different types of structural components.